Hello, my name is Rosa Mendez. I'm a senior at Westminster College, and I will be presenting on the regulation of chaperone mediated autophagy by pharmacological activators and inhibitors. This research was done under the mentorship of Raj Ghosh at the University of Utah and with the help of the McNair Scholar Program at Westminster College. So first of all, what is chaperone mediated autophagy? Well, chaperone mediated autophagy, or CMA as I will be referring to from now on, it's an intracellular breakdown system, meaning that within the cell it breaks things down. In this case, we're going to break down protein that it's either unhealthy or redundant or it's just of not use of the cell into amino acids, which again, amino acids are the building blocks of protein. There are two main parts to CMA. One is Hsc70, which is our chaperone molecule, which acts just like a chaperone. It binds to the unhealthy protein and kind of just walks it down. This unhealthy protein has to have a very specific amino acid sequence, which must be KFERQ. So within the protein, it must have that sequence. And it walks it down to our second important part, which is LAMP2A, which is the door to the lysosome. And just as a quick reminder, lysosomes are organelles within a cell where things get broken down. So LAMP2A recognizes Hsc70 and it opens to allow the protein to get into the lysosome, where it will get destroyed into amino acids or recycled into something else. So why is CMA important? Well, CMA is protective of various diseases. It will either go faster or go slowly or more slowly depending on the disease. It has been linked to diseases such as hepatitis B, neurological diseases like Alzheimer's, cancer, and COVID-19, which are all of our ongoing research to know how CMA interacts, but also how can we do something to help it. So even though we know how these diseases are linked to CMA, there's not a lot of methods to monitor CMA activity. That means that we don't know how much of these unhealthy proteins are actually getting destroyed by CMA. So our question is, how can we regulate chaperone-mediated autophagy using pharmacological agents? We wanted to use pharmacological agents because these are available to all scientists, and we didn't want to just produce something that will activate or inhibit it, as we want this to be accessible to other scientists. Our hypothesis is that we hypothesize that the pharmacological activators or inhibitors used to manipulate CMA pathway can serve as an effective tool to study the clearance or recycling or destroying mechanism in different disease conditions. This means that what we learn from our research will serve other scientists that are actually studying how to cure or treat these diseases to use CMA as one of the pathways that they could potentially upregulate or underregulate to prevent, cure, or treat diseases. We first chose our inhibitors. All of our inhibitors interact with Hsc70, which means that this step will not happen or will happen slower at a slower rate. So when it will, so LAMP2A and the destruction of the protein will likely not happen, or it will happen at a very slow rate. And then we have our activators, which upregulate the whole mechanism. We chose two activators, both these two instead of acting with the chaperone molecule, they act with our DOR molecule, and they kind of increase how much substrate it can take, so it makes it faster. So there will be higher levels of our chaperone molecule as well as our DOR molecule. So what did we do? We obtained and grew cells. We first grew endothelial cells, which are cells that surrounds your blood vessels, and then we have cardiac cells, which are your heart muscle cells. Then we treated our cells at different concentrations, and then we intended to treat them at different times. But due to the craziness of this year, we only had time to do it for 24 hours. And we also only had time to do one activator and one inhibitor. For our inhibitor, we use BER 155008, and I will just be calling it BER for now on, in which the concentrations that we selected were previously tested on Hsc70, and it was just the Hsc70 chaperone molecule, so we really didn't knew how it will interact in a cell level. And then for our activator, we chose humanin G, which was previously tested on rat liver cells. And these are the concentrations that we came up with. We had 2 plates for our endothelial cells, and we just tested humanin G on them. And then we had 4 plates for BER, and we tested, for our cardiac cells, and we tested both the activator and inhibitor. As you can see on this plate, we put 1 concentration in each row, and then so we had 3 wells for 0 millimolar, and then 3 wells for 5 millimolar, and so on. So once we treated our cells and incubated for 24 hours, we collected the sample. So we grabbed the sample and put it in a test tube, and it was 1 for each well, not 1 for each concentration. Then we sonificated to just break down all of the cells walls so we could further see what proteins had the cells. Then we used a Bradford assay to know the concentration of protein, and we looked for the lowest protein concentration, and we normalized all of our other proteins to have that same concentration so that all of the levels of protein will be about the same. And then we did Western blotting. And Western blotting is a technique that allows us to see a specific protein. How do we do it? We run a gel, then we transfer that gel into a nitrocellular membrane, which looks kind of like a paper, but it's not a paper. Then we block it using a primary and a secondary antibody, and in the case of Hsc70, we use BSA with non-fat milk, and then C-block for LAMP2A. Once we incubated, we imaged the membrane, and then that's ready to analyze. So these are our primary results. From the top part, you can see the actual imaging from our nitrocellular membrane. What this means is that a thicker, more darker bar has higher levels of protein, and then like a thin, more blurry light line, it just means that there are lower levels of protein. And then on this side we have our cardiac cells, and on the other side we have our endothelial cells. As you can see, cardiac cells levels were tested for LAMP2A levels, and then for both the inhibitor and the activator, and endothelial cells were only tested for the activator, but we were able to analyze for both LAMP2A and Hsc70. So for LAMP2A, for both the cardiac and the endothelial cells, one for LAMP2A and the other one for Hsc70, we can see from the image that about 5 millimolar, they have the same, like they have a more darker, thicker line, which just indicates higher levels of protein. We're not going to talk about the imaging for LAMP2A for endothelial cells treated with humanine. That is because it was very blurry and very hard to see and quantify. Then for our inhibitor BER in cardiac cells, we were expecting lower levels of proteins, and we can see that at about 10 millimolar. And from the imaging, it's hard to actually tell which levels are which, so we have to quantify the data using a computer program. And again, as you can see from cardiac cells and endothelial cells using our activator, we can see that both at 5 millimolar, there's an increase of the levels of both LAMP2A and Hsc70. Then with BER, which is our inhibitor, we can see that for cardiac cells at about 10 micromolar, there's a decreased levels of LAMP2A. So our results are favorable. They look very promising. As you can see, our inhibitor seems to be lowering the levels of the proteins, but and our activators seem to be upregulating the levels of our proteins again. However, we need to normalize our data to actually make any conclusions. As you can see, there's kind of some curvatures in our image. That just means that there was some error within the loading, so I might have made a mistake when loading the gels, which just tells you that when we normalize the data, this might just say that we actually didn't get any data, which will suck, but it's okay, it's just science. We can also look forward to analyze the media to quantify if cells are being killed. So the media is just where we feed and we grow cells, and we want to analyze the data for one reason. As you can see from this graph of in cardiac cells, the levels of LAMP2A for both 0 millimolar and 20 micromolar, are the same, and this can just indicate that we were killing cells. And we think that because DMCO, it's toxic in cells, so they might just be dying. So we will also have to analyze if it's worth killing our cells in order to inhibit CMA. And we will also want to explore the other inhibitors and activators that we didn't have time to do so that we can make actual conclusions or what are the best methods to monitor CMA. Thank you for your time and thank you for listening to me. I hope this was of your interest and it was informative. I will first like to thank my McNair program, especially Joe, Vanessa, and Healy, because they allowed me to work on this project. And I will especially like to thank Joe, which is retiring this year. I would like to thank the University of Utah, especially Raj, which guided me through all of this process, the John David Simmons and CM Budina for allowing me to work in their labs during these crazy times, and especially for allowing me to work with them even though I'm an undergrad, and all of the lab members. And if you have any more questions, my email is here and you can feel free to email me. Thank you so much and have a good day.